Hello, everybody. This is going to be an overview, as I said, on the TB Oxford Nanopore sequencing pipeline that we've been developing at UCL. The paper is currently on Biomed Review, and it has been accepted at PLOS Global Health Medicine. So that should hopefully, the full one, peer-reviewed one, should be coming out in the next couple of weeks. What we wanted to try and do was develop a whole genome sequencing sort of pipeline to be used to complement routine diagnosis in resource-constrained countries or settings, especially to try and understand hard-to-treat cases such as relapse versus reinfection or different drug resistances. And what we aimed to do was build a pragmatic pipeline. So what I mean by that is that we wanted to try and balance the cost of the pipeline, because obviously if it's too expensive, nobody's going to want to implement it, with accuracy, because obviously if it's not accurate, then it's not going to be a very good research tool or a diagnostic tool. And then sort of user friendliness, because we assume people from various different sort of parts of research and science and healthcare are going to be using this, many of which might not have sort of line command experience. So we tried to make it as user-friendly as possible, especially in the bioinformatics sort of side of things. And also, hopefully, it is as quick as we can make it. Moving, there we go. So Oxford Nanopore is a specific sequencing platform. It has a cheap initial setup. So a starter pack costs about $1,000, and when you compare that to, say, Illumina, which is more like, I think for a MySeq is something like $50,000, it's much cheaper to set up. It has a much smaller laboratory footprint, and if you can look in the bottom right corner, you can see the lady with the laptop. What she is pipetting into is a MinION, so it's very small, it's very portable, as whoever came to the training session that we ran in 2022 will know that I bought them all in my suitcase. So they're very small and portable and easy to set up within a lab. It utilizes long read technology, so that's can be really useful for spanning difficult to read sequences, and TB is full of them. It also has a relatively straightforward library preparation. The kit that I am going to kind of explain to you, and that ended up in the final pipeline, it takes maybe around 2 hours or so to do the actual library preparation, so it's relatively quick, and it's also relatively straight. So how Oxford Nanopore Technology sequencing works, as I say, it uses long read technology, so that's strands of DNA in the thousands or hundreds of thousands, whereas something like Illumina tends to use a couple of hundred base pair strands. DNA moves through a pore on a charged membrane, and as each base passes through that pore, it creates a unique change in the charge. And hopefully this should explain it a bit better. So this is your DNA with the green blob on it, and it's going through the blue pore on the membrane. Every time each of those bases passes through that pore, it creates in the grey box, it creates the squigglegram. And then you use a Basecalling tool such as Guppy or Dorado to then change those squiggles into the A's, T's, G's, and C's that we as humans can read. TB is not without its problems, as I'm sure everybody knows. I assume everybody works on tuberculosis. It's very slow growing, so it's not very easy to diagnose in a time-friendly manner. Often when you extract it, I imagine most people possibly use CTAB, you get quite a low DNA yield because it's got its complex cell wall structure, and it makes it quite difficult to break the cells open without damaging the DNA. And the genome has a high GC content and many repeating regions, which makes it difficult to process and also then sort of analyze further downstream. And then if you are working from a resource limited laboratory or country, there are extra logistical challenges with sequencing, but I will explain a bit more about that further in the presentation. this is kind of the pipeline and what I wanted to get out of the pipeline. So the first is obviously growing the DNA and extracting the DNA. And you need to kind of balance the speed of getting the amount of DNA that you need versus the actual amount you need, because it's very important to get good quality DNA. And I'll explain more about that later. And also the availability of the extraction and growing techniques that people might have in their different laboratories, because obviously you can't do techniques that you don't have in the lab. In terms of library preparation, there are different library kits with Nanopore, and it's important to try and balance the input DNA. So some of the kits that give you more data require more DNA in the first place. So as I was saying with TB, sometimes it's quite difficult to get high yields. So it's a balance between the input DNA and the output data. Then with Basecalling, so in this project we use Guppy, but Dorado I think is now sort of overtaking that. We wanted to try and balance accuracy and speed, and this is very much down to computing power. And then in the analysis as well, it's also sort of accuracy and detail versus how easy is it to use. The majority of people that might be using this pipeline probably don't have a lot of command line experience. So we tried to test out different browser-based options. So for the extraction, we tested out CTAB. So generally, CTAB is a precipitation-based method. But what we were finding when we were sequencing is that we think there are inhibitors being precipitated alongside the DNA. We don't find that in other sort of types of bacteria. So we're thinking maybe it's coming from the mycobacterial cell wall or the mycolic acid, something like that. And these inhibitors, we believe, are stopping the transposase binding to the DNA, and this reduces the number of reads going through the pores. And what I mean by transposase, the way the native DNA kit works is you have these sort of orange balls, protein balls, and this comes in the kit, and they have these barcodes attached to them, the little red strands. So when you combine your genomic DNA with these transposases, what the transposase does is it goes along, attaches to the DNA at a completely random place, and then breaks it in half. And at the same time, it adds those barcodes to the end of the DNA. Now, if there are things being that are inhibiting the addition, sort of the attachment of this transposase complex, then actually your DNA won't be sort of prepared for sequencing. It will just remain DNA, and to be sequenced, it requires the attachment of these adapters and things. So we believe that with just normal CTAB, with precipitation-based CTAB, we were finding that things weren't binding properly to the DNA. We also find that there's a low DNA yield from midget tubes. So what we ended up doing was using 7H11 plates and sort of scraping pretty much the entire plate off to get the optimal amount, or to get as much as higher concentration as possible. And again, I'll explain more about concentration and DNA quality a bit later on. So what we wanted to look at was Oxford Nanopore had been developing a spin column based CTAB method to see whether that improved the number of sequencing reads. This table, which is in the paper, so in blue it's precipitation CTAB, and in green it's spin column CTAB. So this is concentration, and you can see that precipitation CTAB does provide you with more DNA than the spin column one, because you co-spin with quite high salt, and that reduces the amount of DNA, but it does improve the sequencing. So what we found was that the spin column, so the midget tubes were not, did not provide as much yield as we needed. So what we ended up doing is in the light green is spin column CTAB and using a 7H11 slope. So the DNA yield for the kit we ended up using is 5 to 44 nanograms per microliter. So that provided enough. In terms of size, Oxford Nanopore sequencing, as I say, is long read technology. So it's actually very important to have good quality, large DNA. And so again, we found that the midgets for the spin column weren't providing enough in terms of the size. They were too small, but actually all of the other methodologies were okay in terms of the size. So what we were finding when we were doing this, testing out the different sizes, was that somewhere between about 30 kb and 50 kb from your extraction was sort of the ideal size for your DNA to sequence properly. I just want to sort of step back from that though and explain a little bit more about the importance of good DNA. What we found is that it's best to aim for the maximum concentration that the kit suggests, because obviously the more DNA you put in, the more sequencing you will get out of it. So for the rapid barcoding kit, the native barcoding kit, and this is the R9 version, there is now the R10 version, but this kit requested 50 to 100 nanograms, which works out to be somewhere between 5 and 44 nanograms per microliter. If you aim for sort of 44 nanograms per microliter, you will get much more data out of your sequencing. It was also really important to try and get all of the samples to have approximately the same molecular weight, because if you had some with a low molecular weight and some with a high molecular weight, the one with a high molecular weight would out-sequence the one with low molecular weight, and that one, the low molecular weight one, might have to be sort of repeated. So I found it was really, really important to try, and if you had, say, 25 samples that you wanted to sequence, if some were 15 kb and some were 60 kb, I would run all of the 60 kb ones together and all of the lower ones together, because that 15 kb one you won't get very much data from. Something that a lot of laboratories ignore and don't do is check the size of the samples. So you can use TapeStation, you can use bioanalyzer, or you can use sort of traditional gel electrophoresis. But I would say not only is it important to get the concentration of your sample, but it is so, so important to get the size of your DNA. And it's very much rubbish in, rubbish out. And you will find this if you start doing it and you ignore doing the size analysis, you will find that your data isn't very good. So here, for example, this is a readout from our TapeStation, so that's an automated gel electrophoresis machine. You can see that we've got a number of different samples. Some of them look better than others. So this one here looks really nice. It's a very neat, tight band.
Sorry.
Yep.
Should we wait for you to finish before we can ask questions, or we can just?
Well, I mean, you can ask a question now.
Okay. Yes, you have been speaking on the difference in molecular size of the samples.
Yep.
Yeah. So you will need maybe to like optimize the concentrations between the samples to make sure that they are nearly equal before you can run them. Or what can you do now? Because if we expect to have samples that are having different molecular side of the DNA.
Yeah. So ideally, as I say, TB can be difficult, but if you are doing a batch of CTABs, generally they tend to come out sort of roughly the same. But I would recommend, so we tried doing, so you can normalize it to femtomoles, so the number of bases for each of the samples. So there are various calculators online where you can work out sort of the femtomoles, but actually really the best way of doing it is just to ensure that all of them have the same or roughly the same molecular weight. So if you had one that was 15, it might be worth re-extracting it so that you can see some of them just don't seem to want to be a higher, molecular weight. It would be, or if you've got lots, it would be worth running all of the ones that are, say, I don't know, 40 to 60 kilobases together, because they should all sequence roughly equally. And then if you had another lot of samples that were, I don't know, 15 to 30, then you could run those together. But if you, as I say, if you ran one that was 60 kb and one that was 30 kb, that is, if you think about it, sort of, half again the amount of DNA. You've got double the DNA in that 60 kb one, and so you're always going to get twice the amount of information out of it. Does that make sense?
Sure. Yeah, sure. I think we also need to have the standard difference in concentration between samples before, because someone can load same a pool of sample with the different concentrations.
Yes, you should also always normalize all of your samples. So what you tend to do is normalize it to the lowest concentration samples.
Okay.
Excuse me. So if you have one that's 5 nanograms per microliter and all the rest are 40, you would have to normalize them all down to 5, or alternatively, I would remove and repeat the one that's 5 and do everything at 40, because you're going to get much better data if you do them all at the higher concentration. So basically, it's very important, especially with TB, it's really important to get as good a quality DNA as possible. You can sort of get away with not being quite so accurate with things like E. coli, but TB, because it's such a complicated organism, it's really, really important to get this really good DNA. I mean, test it for yourselves. If you put rubbish in, you will definitely get rubbish out. So it's well worth spending that extra time, I think, on the extraction, being very gentle with the extraction, because again, you want to try and maintain these really long strands. So not pipetting aggressively, being very gentle. And then, yeah, definitely doing concentration, but also doing this size analysis as well. As I say, it takes, an extra hour or so to run a gel. It's very much worth it, I think, in the end.
Sure. Thank you.
So this is the automated gel electrophoresis, and you can see the one that I've highlighted in red is nice and dark. So it's got a lot of DNA in it, and it's at the top of this ladder. So I think this ladder goes up to 60 kb. So this is probably 60 kb. All of the strands are roughly the same size, and it's all in a very neat kind of band. Therefore, all the DNA is fairly good quality. Whereas if you compare it with this one, you can see it's much more of a smear. It's further down, so the sizes are much lower, but it's also just a load of smear. So you'll have some strands that are, maybe 30 kb, all the way down to in the hundreds of base pairs. So I would run the first one, the one in D1, but I definitely would not run F1, because even if you run F1 with a load of very good ones, it tends to mess up the sequencing. So it's really, really important, and it's much more cost effective to do these extra steps before you sequence, because the sequencing is always going to be the most expensive part. We can then move on to library preparation. So as we were saying, TB extractions generally yield very or relatively low concentrations, and it's not enough for something like the Oxford Nanopore ligation library preparation kit, which requires 1 microgram of DNA. So we tested out 3 different kits. Now, again, as I say, these are all the R9 kits, but the R10 kit, the version 14 R10 flow cell kits are now available. We tested the PCR based kit, because you can add in less than 2 nanograms per microliter of DNA. But what we found when we looked at the sequencing, we visualized it, we found that amplification wasn't even across the whole genome. So this is, hopefully you can see, this is RPOB, and you can see that half of RPOB has some reads, those black lines at the top, and the other half of RPOB has nothing. So sequencing with PCR, whilst it does amplify the DNA, it doesn't amplify it very evenly, especially for TB, which obviously has, as I say, these repeat regions, these GC-rich, this GC-rich genome. So we scrapped that. We decided that wasn't good enough. We tested the rapid barcoding kit, and this is from native DNA. So that is, you extract your DNA and then you directly prepare it for sequencing. There's no amplification involved. So this was the original version of this kit. You needed 400 nanograms of at least 30 kb DNA. And again, we tried to normalize it to femtomoles to try and make the barcode reading more even. It did have better coverage, but it definitely needed improving. You can see that we've got quite a few more reads than the PCR one, but it still wasn't really good enough. Then this rapid barcoding kit 96 came onto the market. You needed less DNA, so 50 to 100 nanograms. and actually it sequenced far better. So within 24 hours of sequencing 6 isolates, we had more than 40 times depth, and after 48 hours, all of them were much, much higher than that. So generally it's sort of suggested that 40 times depth is the minimum you would need for nanopore sequencing, but again, you would need to validate that for your own laboratories. And this is what we found with this 96 kit. So you can see that there's far more reads across all of the area of RPOB, so much better coverage and also much better depth. And as I pointed out, actually now, since we published this paper, the R10 version flow cells and the version 14 kit chemistries are now available for these kits, and they will be much, much better. So you should definitely buy, well, in fact, I don't think you can buy the R9 and these kits anymore. So all of the kind of results that we got here should actually have been improved massively now with this new chemistry. It's much more accurate. In terms of Basecalling, so we used Guppy, but as I say, there are various different versions, and I think now that Guppy is also being replaced by something called Dorado, it's just another Basecalling model. Within these programs, there are a number of different Basecalling models. So there's fast, and as the name suggests, it is the quickest version, but it isn't the most accurate. You then have high accuracy Basecalling and super high accuracy Basecalling. And obviously, the more accurate they are, the slower they become. We tested these different models on a fairly sort of bog standard sequencing computer. So it didn't have a GPU, it just had a central processing unit, which I think was 32 gigabytes. So much more powerful than your sort of standard laptop, but not sort of really, really powerful. So what we found was that for each of our FastQ files, we found that obviously fast was much, much faster, but you can see with the Q score and the accuracy that actually it was lower than the high accuracy and the super high accuracy. What I found when I tried to run super high accuracy on that computer was that you can see here that I've put it took more than 36 hours to run, but actually it took sort of about a week for it to do super high accuracy Basecalling on the same file. It took 50 minutes to do using fast Basecalling. However, if you have a very powerful computer, and this is if you are setting up a sequencing sort of laboratory, getting a really powerful computer is going to be really, really important. You can do high accuracy Basecalling in real time as you're sequencing. So they are coming down in price, GPU, so graphical processing unit computers. And I thoroughly recommend buying the best computer that you can afford because sequencing technology is improving so quickly, your computer will very quickly be obsolete, and it will no longer be able to keep up with the power that sequencing needs. So I would thoroughly recommend buying as fast and powerful a computer as you possibly can, because otherwise you'll just have to buy another one in a couple of years because it won't be able to keep up. So we would recommend high accuracy as a kind of middle ground. We found that high accuracy and super high accuracy were both called all of the SNPs that we were expecting. Fast was almost as good. I think it just missed one. But obviously it does take much longer, and then you have to take this into account if you are doing this for a patient who is waiting for appropriate treatment. So again, it's something to consider when you're putting this into your laboratory. In terms of analysis, there aren't many non-coding programs that will take Oxford Nanopore sequencing data. I could only find TB Profiler and Microbe. And what we found was that TB Profiler had both a better database, so a wider database, and covered more drugs, drug SNPs rather, and it was also more accurate. So when we compared Microbe to TB Profiler, we found that it wasn't picking up as many of the SNPs as TB Profiler was. So we decided to take TB Profiler forward. So this is a web browser based version. So there is a command line version, but there is this TB Profiler web version. So I can show you how this works. So this is the web page. It says, I've added the link to the presentation, but I can also send it to you as well. Step 1 is add your sample. So you just press upload, and here you can drop your files into it, and you just have to make sure you change it to Nanopore and single read, because Nanopore only uses, it only sequences one strand. So let me just put one of these in. You'll see it will upload it very, very slowly. And once you have uploaded it, as I can't be bothered to wait for this to do anything, what it will give you is a sort of long, complicated number, and you can then check back. So if you have a look on the home page, hopefully it'll go back, you can view your results. So when this uploads your sample, make sure you save that long number, because then you can put your sample ID in, submit it, and you will get the results. So this is the number, for instance, that you will get. TB Profiler, depending on your internet, might take a while, so it's definitely worth saving that number and maybe coming back to it later. For me, we're using UCL internet, which is really fast. If I run one sample, it probably takes about 20 minutes or so. So it really depends, I think, because it's a browser-based thing. It depends on your computer and your internet. So this is the summary of what you will get out of your sample. So you can see it tells you what lineage you've got. So this is Beijing, and it will tell you what drug resistance it thinks it is. So it has decided that this is MDR-TB. And the reason it has decided this is because of the drug resistance SNPs that it has found in your sample. So it also has, so this is the online report, but you can also save it in an Excel table or as text or as something called a JSON file, which you can view online. I would download. Sorry?
Work offline, mate. Can the TB profile also work offline, because you find some issues with the internet?
Yeah, so there is a command line version, and you're more than welcome to use that, but you will have to understand the command line. I can show, well, you can, it's fairly easy to download. It uses Ubuntu, so it uses the Unix sort of system. But obviously, if you don't know how to use command line, you'll have to learn how to do that. So yes, there is an offline version, but it does require some command line knowledge, which is why. Sorry, yeah.
Okay. Yes, additional question. Will this also be able to identify mycobacteria other than tuberculosis, because we see the prevalence is growing, and we think it's something that we should also pay attention to?
Did you say non-tuberculous?
Yes.
Yeah.
Sure.
So this is currently only TB, but I know that Jody Phelan, who created this, is working on an NTM version of TB Profiler. If you were looking for something that, or if you were looking to check if it was TB or not, there are various other online web-based programs that you can use that should be able to speciate for you. So there's something called the Center for Genomic Epidemiology, which, let me show you. So this is quite a good, I mean, there are various different things, but this is quite a good service. It provides lots of different programs. So if you go on to Kama Finder, it predicts the bacterial species based on, I think it's the NCBI database. So you would leave that as bacteria, and then you would just choose and upload your file, and then it will give you sort of a list of the organisms all of your reads best match with. So in theory, if it's a very pure culture, then it will come back with this is TB. If it's not a pure culture, then it might come back with multiple different things. And also, if you have lots of small bits of DNA, it can come back with lots of different things, especially with TB, because obviously if you have a read of 200, say, base pairs long, if you've put really poor DNA in, it's very difficult for the program to identify whether it's TB or whether it's, you know, abscessus, or whether it's, well, whatever, Canetti, because if that specific 200 base pairs is part of that repeating region, then it will be really hard for it to identify exactly where it belongs to. And so you might get a less sort of specific result. But there are, this program, there are various other ways of doing it as well. So you can definitely identify which species it is. But currently TB Profiler doesn't do that, but I do know that Jody is working on an NTM version of that.
Okay.
So yeah. So you've got all of the resistances, and these are the mutations. So this table is based on the WHO's TB drug resistance mutation catalogue. So these are all based on that. So you can see here it's got comments about where they came from, associated with resistance, that kind of thing. There's various other pieces of information that it can also provide you. And I think down at the bottom, it gives you the kind of, this is the genome itself, and you can see the different SNPs. So it's a very useful program. I highly recommend having a go with it. Right, let's go back to that. So that's TB Profiler. Oops, let's share that properly. So yeah, so this is basically what I just showed you. As I say, it tells you the number of reads that you've put in. It tells you the number of reads that it managed to map. So it uses H37RV, I believe. So it managed to map nearly 97% of those reads to H37RV, which is pretty good. The median coverage. So that's, I think what by that it actually means depth, is 194 times. So that's very, very good quality. And then it was able to give us all of this information. These two are just different versions of the output. So in the middle, we've got the text output. It's exactly the same thing, but it just is a text file. So you can save that to your computer. And on the right is the JSON file. I don't like the JSON files very much. I find that they're not very easy to navigate, and you also have to open them only in Firefox, or at least I seem to only be able to open them in Firefox. So I would always save it as a text file because it's much easier. Obviously, you can read the report on the website, but the report will disappear after so many days because they delete the data, and obviously because more and more people are using it. So yeah, I would save it as a text file. This then is the sort of final pipeline as we see it. So for the extraction, we wanted to test speed versus concentration and sort of DNA quality. And we found that so column-based CTAB or a validated extraction kit, if you are, if you have something like QIAGEN. I don't think, having tested it, QIAGEN will not give you enough DNA. So CTAB or column-based CTAB, I think, is the best method. We have added the column-based CTAB protocol into the paper, and it's also in the lab manual that we've created and put up onto the website. In terms of library preparation, again, it was a combination of the amount of input DNA versus the amount of data to get out. And we found that the rapid native barcoding kit and what will now be the version 14 kit was the best version for that. In terms of base calling, accuracy versus speed very much depended on computing power. High accuracy base calling would be the kind of best middle ground. I mean, super high accuracy would be ideal, but fast base calling, if you don't have a particularly powerful computer, would be an option, but you just have to, validate that and make sure. It would be even more important to get very good quality DNA and a high number of reads to make sure that fast base calling had enough data to work with. Then in terms of the analysis, accuracy and detail versus ease of use, TB Profiler definitely came out on top. Whether you use the browser-based version or the command line version is obviously entirely up to you. Here in the UK, well, certainly at UCL anyway, we have very good internet speeds. And so if I'm just doing something very quick, I just put it onto the browser. The command line version gives you options for different inputs and different outputs. So you can slightly edit what you get out of it. So if you are interested in learning how to do that, it's worth having a look on GitHub and those kind of pages. And again, I can send you the link. This then is the kind of final pipeline in its complete form. So you start off with inoculating a 7H11 slope or plate so that you can get as much bacteria and therefore DNA as possible. We found that incubating it for 2 weeks, so I put post-flagging, and that's based on our midget cultures. So we put the same amounts of culture or sort of frozen stock onto a 7H11 plate and a midget plate. And we found that sort of, I think it was 3 weeks in total was the best. It gave the optimal amount of growth. Then extracting it using this column CTAB method, and then making sure that you had between 50 and 100 nanograms of at least 30 kb DNA was the best amount of DNA to use. Then using the native library preparation kit, we ended up sequencing about 12 samples on a flow cell. This again was the R9 version of the flow cell. With the R10, it will be more because it is better. So for the R9 with something like E. coli, we can run maybe 20 samples and still get good enough depth. With TB, because it is much more complicated, we're currently running 12 samples on a flow cell, but I know that there are people who have been using the R10 kits for TB in their labs, and I think they're saying they are using closer to 20 samples per flow cell, which will obviously decrease the cost per sample, but you do have to balance that with the amount of data that you're getting out of it. Then high accuracy base calling or super high accuracy base calling would be ideal, and using TB profiler to analyze. But as I have said, and it's very, very important, each laboratory is very different. You might have a midget machine, or you might not have a midget machine. You might have access to CTAB, or you might not. You know, you might have a very powerful computer, or you might not. So it's really important to validate this pipeline in your own settings. We have kind of done the background legwork, as it were, but it's really important once you set it up in your own laboratory that you are sure that it works in the way that it's intended, using the kit and stuff that you use yourself. A quick word on logistics, because I've done this in various countries now, and some places are harder to sort this out than others. Lead times and customs issues can be a real problem. Nanopore, if you're buying stuff directly from Nanopore, they are quite good and they do ship relatively quickly, but getting stuff through a country's customs can be problematic. I think, if I remember, Juma, it took about a month to get them into Tanzania when I sent you these things. I can't remember off the top of my head. In Zambia, it takes about a month to get it through customs, and Gabon. Some countries are very quick, some countries take a long time. So it's very important when you're considering the use by dates of these items. Obviously, you presumably have these things to consider already with some of the kits. But the flow cells, in theory, have a warranty of about 3 months. What I find is that as long as they're kept in the fridge in an appropriate temperature, then actually they do last for much longer, but the warranty is for 3 months. It's really, really important to store the reagents and flow cells at the right temperature. I cannot stress this enough. Do not freeze the flow cells. If you freeze the flow cells, you will destroy all of those pores, because they are originally mammalian pores. They do not like to be frozen. I went to one country and I'd shipped stuff, and they had misunderstood my instructions, and they froze 5,000 pounds worth of flow cells, and they were all dead. So please do not freeze the flow cells. You can freeze the reagents, check on the box, it does say, but do not freeze the flow cells. You will need power for the duration of your sequencing run. And if it does cut out at any point during that run, it will save the data up until that power cut. So if you have UPSs, it's well worth attaching it to the computer. Sometimes using a laptop, if you have a powerful laptop, might help, because obviously you've got that battery power until the mains come back on. But yeah, these can run to 72 hours. So it's really important to try and have a secure power supply for the entire time. Equally, you do also need an internet connection. And as I'm sure if you were there in Tanzania, you remember, we really struggled to get the runs to start because the internet wasn't good enough. I think it took about 6 attempts to get it to start. And this is because as soon as the run starts, your computer is pinging information backwards and forwards to the Nanopore server. It sometimes downloads things, especially if it's the first time it's been used in that area. So it's really important, especially right at the beginning, to have an internet connection. Ideally, you would want an internet connection throughout, but I think it doesn't use the internet at all times. I think it just pings back up and down to the server. So ideally, also a stable internet connection throughout, unless you have a Mark 1C version. So that's the MinION with a small computer kind of attached to it, a bit like a tablet. That does not need the internet to run. So if you do have very difficult internet, then it might be worth investing in a Mark 1C, because it also has a very powerful GPU sort of computer in it. So it's more expensive, but it probably is worth it if you are struggling with the internet. Storage is also an issue that a lot of people kind of forget about. So just one MinION flow cell can generate as much as 200 gigabytes of data. So you really need to consider storage and where you're going to put all of this. Your computer itself will need that amount of, or at least that amount of space on the C drive. It's not recommended to immediately store that information on an external drive, because you might lose connection. So once you have sequenced, it's on your C drive, then you're going to have to find somewhere to put it. So we here at UCL use remote storage, sort of cloud drives, where we keep all of the data. But you have to consider where you might put it. You're going to get through quite a lot of hard drives, because I think within 3 or 4 runs you're going to end up with at least sort of a terabyte's worth of data. So that is something very much to consider, and it also can get quite expensive to store that data. You can delete the raw files and just keep the FASTQ files, which will reduce the amount of storage. But you might want to go back to those raw files as Basecalling improves, because you might find that you get better quality data, but obviously that's entirely up to you. I've explained how it works and how you can go about doing it, but if you want to learn more about the pipeline, and this is where I'm hopefully going to put this presentation, we have these Pandora Global Health Network Hub pages, and I will show you. It is, oh, go away. There we go. So on this Pandora page, we've got a sequencing tab, and within this sequencing tab, we've got a number of sort of child pages. People might have been on these before, but we've put a lot of sequencing tutorials online. So how to QC a flow cell, loading flow cells, washing flow cells, that kind of thing. So these are all sort of general Oxford Nanopore tutorials that we've made, but we also have a specific page for the tuberculosis sequencing pipeline, which is what I've just explained. So you can see here that the med review paper is, there's a link to that there. Hopefully I will be able to exchange that for the actual peer-reviewed one when it's finally published. But there's various different pages on culturing, DNA extraction, quantification, library preparation, data analysis, etc. We've also added a page on costs. We didn't add that to the paper, but we've added a page on costs just to help you identify how much it might cost per sample. And we've also added a laboratory manual to the website. Hopefully this will open. Yep. So this is the paper, but in much, much more detail. So this explains exactly all of the, all of the methods, how to do all of those different bits, and the sort of principles behind it. So hopefully you can download that, and that should be enough to explain how it all works. We also ran a sequencing workshop in Zambia, I think the February after we did it with you guys, and I actually recorded all of those presentations. So again, it goes into more detail than this presentation about all of the different sections. So an introduction, how to do CTAB and the importance of CTAB, why TB is so difficult. I feel like that's a common theme. And then the library preparation, how to use the rapid barcoding kit, and then sequencing analysis. So this obviously, as I say, was kind of a very basic explanation as to how to use it. If you are interested in learning more, then you can go through and watch these videos, have a look at the laboratory manual. if you're interested in learning command line, you can look on this bioinformatics page, and it will kind of explain the best places to go to start learning. There are loads of online sort of tutorials and, you know, YouTube videos that will explain how to learn bioinformatics and using the command line. I think in science now it is really important to learn, so it's well worth getting used to it. I think I was and have always been a sort of wet laboratory scientist, and I taught myself completely from scratch in about 6 months to do the very basics. So I think I'm not really computer literate particularly, but I must admit I'm very pleased that I have learned it. So it's, if you have the time to learn it, I think, you know, it will really help in your career as well. So I recommend having a look at these pages as well, and it should help you understand how to do that. I think that's pretty much it. Yep. So this is just thanking everybody who has helped me with the protocol, actually setting it up. So yeah, I think that's pretty much it. Does anybody have any questions? I'll leave that on that.
Yes, I have a question.
Sure.
Okay, for the culture media, you use 7H11 slope.
Yeah.
For us, we literally use LJ.
Yeah.
So I was wondering if LJ was also tried.
Yeah, we did try the 7H11. That also works. It's very much solid media versus liquid media more than anything else. For research, we tend to use 7H11 because it does give you slightly more, we found anyway, slightly more growth than the slopes, purely because we buy in our LJ slopes, whereas I can make up a whole plate of 7H11. So if you have LJ slopes and you can get enough bacteria off to get the amount of DNA that you need for the kit, then by all means use LJ slopes, yeah.
Okay, and now how long does it take from extraction to now having the result?
So with extraction, so with the precipitation CTAB, it takes sort of about a day and a half, doesn't it? With the column-based CTAB, it does take less time, so you can do it in a day, and in fact you could probably do that and the extraction, the quantification in one day. So the extraction and quantification might take, say, a day. Sequencing with the library preparation, it might take you the morning, and then you can run that sequencing, I would say run it for as long as possible, so 72 hours. So if you were extracting on Monday and then you started your library preparation on Tuesday, by the end of the week you should have your data. And then, so the issue is obviously with this power of the computer, you can base call at the same time as you sequence if you have a fast computer that can cope with it. If you do not have a fast computer that can cope with it, I would sequence and then I would base call it separately afterwards, because you want your computer to be focusing on sequencing. The reason that graphical user interface GPU computers are much better at this, I don't know if anybody games, but the gaming sort of, if you are in a gaming world, you've got a lot, the computer has to deal with a lot of tiny bits of information. As you move around that world, everything changes sort of in very, you know, minute detail. Graphical user interfaces are really good at processing thousands and thousands of tiny bits of information at once. CPUs, central processing units, which most computers have, aren't so good at that. Sequencing works very similarly to gaming. So you have lots and lots of thousands and thousands of tiny bits of information that your computer has to process all at once. If you are making it sequence and also making it base call at the same time, you are making it do two things at once, and it won't do, if it can't cope, it won't do either particularly well. So Nanopore has a page on its website about the IT requirements. As I said, I would try and buy the best computer you can with the money that you've got. So we bought a GPU computer last year, and I think it cost us £2,500. I know our group in Zambia that we work with, they bought one. Again, I think it was last year, and I think it cost them about £3,500. It obviously depends on the country you're in and what specs you are buying. But I think sequencing is obviously a relatively expensive thing to do. £3,000 or whatever is actually a very small outlay in the grand scheme of things. So I think it's very much worth buying the best computer you can. So if you are sequencing and Basecalling all at once, that will take sort of 3 days. So and then the analysis, you will have your sequencing data, and then it's just a case of you uploading it or running it using the command line, which again, depending on your computer or your, the internet, might take a day, depends how many sequences you've got to run. So realistically, from extraction all the way through to results could take as little as a week. Does that answer the question?
Yes, thank you.
If you had a slow computer and you were trying to do super high accuracy, it might take about 3 months because the Basecalling is so slow. So yeah, it's very much dependent on what facilities you have in your laboratory. Does anybody else have any other questions?
I'm trying to think about using CTAB for this.
Mm-hmm.
Yeah, because as we know, CTAB is beating,
mm-hmm.
and as we are validating the pipeline for clinical use, I hope so. I think using CTAB can be a bit difficult for
what are you currently using?
Yeah, for in Tanzania we extract DNA using the Genolyze from Hain Life Science.
How much DNA? What concentration do you get there? It's not very much, is it? Because it's only really designed for PCR. This is why CTAB is really the only way to get enough DNA and good quality DNA. So generally, because TB is such a complicated organism to extract, the regular kits that you would just use for, as I say, the gram negatives, like the Sigma Genolute or the QIAGEN ones, or even the Hain one, they're not designed to. They can't work well enough. That's why CTAB was developed. And yes, you're right, it is very, very painful and tedious. But as I say, you can try it out and see how much DNA you get and what quality DNA you get. But in our experience, CTAB or the column-based CTAB is the only way to get the amount of DNA that you would need to do native DNA sequencing without having to use PCR, which then obviously kind of messes it up, as I said previously.
Maybe if the use of CTAB can be automated, it can.
Yeah. I mean, if you've got an automated system, then that's obviously fine. As I say, the only problem with the original precipitation-based CTAB is we found that whilst you get more DNA, you don't get more reads. Whereas if you do the column-based CTAB, which is quicker and easier, you don't get as much DNA, but you do get more reads. So it's up to you which you want to do. The column-based one is slightly more expensive because you have to buy one of the column-based extraction kits to go with it. But ultimately, you get better sequencing quality and you're less likely to have to repeat it. So it's one of those things you in your lab would have to kind of tally up to see which would be the best option for you. Any more questions?
Yeah, so I wanted to find out whether the difference in terms of cost efficiency, if you compare it to the routine standard of care, for example, in Kenya, where we start diagnosis with GeneXpert, then we also go to MISIT, then we also have LPA from Hain Life Science, then we also have first line phenotypic, then second line phenotypic. Yes. Yeah. Have you looked at maybe the issue of cost effectiveness of food?
Yeah, so I know that different places and different countries get different sort of discounts on things like midget tubes and Hain and the GeneXpert. Am I right in thinking that a GeneXpert cartridge is still around $16? Again, I guess it depends on where you are. With sequencing, so we got the sequencing down to around, I think it was about $150 per sample. But with the new sequencing technology that will have come out, you can add more samples per run. And so the cost will have come down. I think the idea of using this pipeline isn't necessarily to do it instead of GeneXpert, but if you are having a complicated patient who isn't responding to treatment, or you're not quite, the midget is not providing conclusive data on what resistances there are, then you might want to then do sequencing because, as I say, it is probably too expensive to do for every patient. So we kind of set it up knowing that obviously sequencing is expensive, although the cost will be coming down, but sort of using it in cases where you can't quite understand what's going on without that kind of granular genomic data. It's also, of course, really useful if you're having outbreaks of something. So for instance, in Gabon, they've been using it to do surveillance of the different lineages across Gabon, and it's the kind of best way to look at transmission. So for instance, if you had never seen Beijing in your country, you might be able to do some sequencing and look out for lineages and things like that. So yes, it's more of a complementary sort of technology rather than a taking over phenotypic analysis or the gene expert.
Yes, I just wanted to ask a question on, because you said during the DNA extraction, you use the column-based setup, and then it will take care of the inhibitors, and probably you said they'll be precipitated out or something like that. Eh?
Yeah.
Then at the end, your product, that is your DNA product, will be of high molecular weight, right?
So the molecular weight depends on how gently you do your extraction. If you sort of pipette it really aggressively, or you sort of vortex it aggressively, then you are going to break down the DNA. So the molecular weight is very much dependent on how gently you treat your DNA.
Okay, and then you say now after you've gotten this high quality molecular weight DNA, so they're the ones that will be able to give you the long reads.
Yep.
Yeah. But then now when it comes to probably now when you're doing the assembly, the sequence assembly, how do you be able to, I don't know if you faced that challenge, probably when now doing your sequence assembly and now with the long reads, because they're normally associated with a bit of errors, or sometimes by the time now you're assembling your entire genome back to its origin, to what you originally had.
Yeah.
Are you able to take care of that during all this time so that you're able to tell that the errors that probably occurred due to the size of the sequence, the template that you're sequencing?
So if I understand the question, you're asking about accuracy of nanopore sequencing, is that right?
Yeah, something of the sort, yeah, sure.
Yeah.
Because it's I think it's one of the only sequence technology that uses the long reads.
So there are others. There's PacBio, there's various other ones, and Nanopore has been aware, and I think they're quite cross now because actually their advances in Basecalling. So you know how I was saying fast versus high accuracy versus super high accuracy. If you use super high accuracy, then it's very, very good. And I think I can't remember what the Q scores were. I think it was 20, which is on a par with some of the Illumina stuff. Also with the new technologies, so with the version 14 kits and the R10 flow cells, it's also improved the Q scores as well. So when you do this with these kits, the Q scores will improve massively. It's also really important to get as much information as possible. So this is what I was saying about depth. So depth is the number of reads that cover each base in the genome. And so the more reads you get per base, the more accurate your data will be. Because if you have 5 bases at a position that is A, and you have 2 that are A and 3 that are T, you're not going to be particularly confident. But if you have 100 bases and 90%, 90 of them are A and the rest of them 10% is T, then you can be far more confident that that base is being called correctly. So that's why having a good amount of data for each of your samples is really, really important. So, and again, this is something that you might need to validate with your own computers and your own kind of downstream analysis tools. But with the new version of the kits, and as I say, with your own kind of QC and that kind of thing, you should find that it is perfectly good for doing that.
Thank you.
And in a way, long reads are better for assembling than short reads, because certainly for problematic genomes like TB, because if you've got a huge span of repeat regions that's, I don't know, 500, 600 base pairs long, if you've only got 300 base pair reads, it can be really difficult for short read technology to place them properly. And obviously, if you've got reads that are very, very similar throughout, and so with long read technology, if you've got a read that's 5,000 base pairs long, then it's going to be much easier to put that in place in relation to the reference genome. It's like filling in a puzzle that's got 5 massive pieces compared to 5,000 tiny pieces. It's much, much easier to put that big piece puzzle together than it is to put the 5,000 piece puzzle together. So yeah, I mean, they both have their pros and cons, and obviously everybody thinks hybrid sequencing is probably the best, but then you have to sequence twice and it's twice as expensive. So they each, long read and short read sequencing, both have their pros and cons. And obviously you are more than welcome to use Illumina. This platform, we designed this to be, so we did compare the Oxford Nanopore reads with the Illumina reads, and they were exactly the same. As I say, the only one that missed one SNP was fast base calling. When you compared high accuracy and super high accuracy base calling to Illumina, when we put them both through, it was exactly the same extraction. And then we sequenced with Illumina and sequenced with Nanopore, and they came back with exactly the same answers. So if you utilize this pipeline, you know, and then validate it against Illumina, maybe for the first few times, you should find that they're pretty much identical, and you shouldn't find that there's any accuracy issues. But again, it's something that you would possibly need to validate in your own site to make sure that you're doing it accurately.
Thank you, thank you.
What is it that that cannot do that Dorado can? Because I have been using that sometime, and I think it's also one of the best calling software that have ever come with.
So I don't. Sorry, carry on.
What the difference? What the difference now?
Yeah, I'm not 100% sure what the difference between them are, but both Guppy and Dorado are nanopore base callers. I have a feeling that Dorado uses more advanced, it's not quite AI, but it's machine learning to improve its base calling. So I have a feeling that Dorado is kind of a more advanced version of Guppy, and therefore does make it more accurate. And I have a feeling that they're now replacing Guppy with Dorado as part of the Minnow software. I updated mine not that long ago, and it still seems to be Guppy. But as I say, I'm pretty sure that they're going to replace Guppy with Dorado. But yeah, I have to keep checking back to see if and when that gets done. I think it would work, I imagine, in pretty much the same way in terms of the coding and stuff like that. Rather, you know, Dorado rather than Guppy as the command. But yeah, I think it is supposed to be just better, because I think it uses a slightly different algorithm to optimize itself.
Yes, I have one question.
Sure.
During Basecalling, what is the minimum time that you need now to get a super accuracy results? Let's say if you're doing it for clinical diagnosis and not research purposes.
So if you, well, again, it would depend on your own sort of situation. But what we found is if you go directly from, say, a midget tube or from a slope, the optimum amount of DNA you will get is after about 3 weeks of growth. And then, as we were saying earlier, maybe about a week to do the extraction, sequencing, and then analysis. So you could try and do less growth. It might take less. I guess it depends on your starting sample. If you're taking it directly from a sputum and you've got to sort of decontaminate it and do all of the sort of that side of things first, then it might take a little bit longer. But in terms of this sort of section of the pipeline, the growing all the way through to the results, we found that you can do it in about a month.
1 month.
Yeah, so not the quickest, but you get a lot of detail from it. And as I say, if you are finding that treatment is not working for a specific person, or something is not quite right, then it might be worth doing sequencing as part of the other sort of routine care that you've got to try and identify, as I say, whether it's a relapse or a reinfection, or whether there's maybe some resistance that you're not sort of you haven't identified as part of phenotypic analysis. Or if you don't do phenotypic, midget analysis, because some labs don't have midgets. So yeah.
From the time of loading in the flow cell.
Yeah.
You said that for high accuracy, you needed sub-accuracy, you needed more than 36 hours.
So with the sequencing, you can set it to run as long or as short as possible. With whole genome sequencing, you will, to get as much information as you can, I would recommend running it for as long as it allows, which is 72 hours. You can run it for shorter, but obviously you will get less data. Generally, the amount of pores that you have that can read DNA will decrease over time. 72 hours is the maximum amount of time you could possibly run it. By then, you tend to really only have sort of a few flow pores actually still running. In terms of the base calling, as I say, so Minnow, which is the program on your computer that runs the sequencing, you can get Minnow to sequence to base call at the same time. So Minnow has, as we were saying just now, Guppy or possibly Dorado now, and it can base call at the same time as it's sequencing. So you could do those two things together. That's if you have a very good computer. If you don't have a very good computer, I would recommend sequencing and turning off base calling and then doing base calling separately, because if you do both at the same time, your computer won't sequence as well. And if the base calling will lag behind anyway, so it will take much longer. This is why it's really, really useful and important to have a very good computer that can keep up, because if you're doing it separately, then it's going to take you however long to then also do the base calling itself. So yeah, as I say, if you have a very, very low standard computer that can only just sequence, if you wanted to do super high accuracy base calling, it's going to take it another like 3 weeks to do it. So it's not worth the effort. So yeah, it's really, really important, I think, to have a computer that can keep up with your sequencing, because otherwise it's going to slow down your time to results.
So when you say a powerful computer, can you be specific, maybe the one that you're using?
So we have, there's loads of different ones, but we have a GPU computer. So it has a specific graphical user interface installed in it. So it's a NVIDIA one. Nanopore, again, if you look on the website, the Nanopore's website, it has IT specifications and it will tell you what the minimum requirements are. I would recommend going as good as you can get for the, the amount of money that you are willing to spend. So I would say at least a computer with a GPU in it. If you have a CPU one, you can probably do fast base calling in real time, but you can't do any, in my experience anyway, you can't really do any more than that. So my laptop, the one that I'm talking to you from now and the one that I brought with me to Tanzania, this is a CPU computer. It has 32 gigabytes of RAM, so it can sequence quite happily, but I can only do fast base calling if I wanted to do it in real time. And even if I did high accuracy or super high accuracy base calling just on its own afterwards, it would still take it ages. Whereas on the GPU NVIDIA computer, so it's a Dell, but it's got a NVIDIA GPU core in it. You can do high accuracy base calling at the same time as your sequencing. It can't quite keep up if you do super high accuracy, but if you did super high accuracy separately, it doesn't take very long at all. So it's very worth getting a good computer. As I say, in the grand scheme of costs, even £5,000 is well worth the effort, I think.
Yeah, then during inoculation, when you're still doing the culture, what's the amount of growth that you'll need to get a good concentration of the DNA during extraction? So just to ensure that you don't have to go back again.
Yeah, so I mean, we inoculated 50 microliters of a frozen stock. So these were all, because as I say, you can't really do it from a a sputum culture because they're all varied. We did them from normalized frozen stock. So we added, I think, 50 microliters onto a midget tube and onto a slope at the same time when we were testing this out. Again, this is all in the paper. And when we left them for varying amounts of weeks. So we allowed them to get to the point where it flagged in the midget tube, which was usually somewhere between about 3, 4, 5 days. And then we allowed them to grow for 2 weeks, 3 weeks, and 4 weeks after that flagging. So we are assuming that the midget had roughly the same amount of DNA bacteria in as the 7H11 slope, because we put the same amount in, and then the midget tube flagged, and then we sort of used them at the same time point. So we found that if you grew them for 1 week post flagging, it wasn't enough. 2 weeks worked quite well, and then any more than sort of 2 to 3 weeks, you started to get death of the bacteria. So we found that the optimal sort of time for growth to get the most DNA out of those samples was sort of around 2-3 weeks of growth. Again, that might be something that you would have to test in your own laboratory to make sure, but yeah, that's what we found anyway. If you're doing it directly from sputum and then you're growing it up, you might have to sub it from the midget that you then, your decontaminated midget, so, or however it is you do it. So you'd probably have to add that extra step in if you were doing it directly from a sputum. Or I suppose if you've done the decontamination, you could then put some of that onto a slope. Again, I guess it depends on how you would want to do it. It doesn't really matter. You just need to get a decent amount of DNA, basically. It's definitely well worth having a practice with some samples, and then also you can use that to validate the pipeline as well, in your own setting. So it's well worth buying a starter pack, just having a go. Your first few ones might not be completely perfect, but you will get better over time. It's not a particularly difficult in terms of the laboratory. It's just a lot of pipetting. It's not really that difficult. The hardest part is loading the flow cell, as I'm sure if you were there you remember. But again, there's the tutorial video online that does go through it. So yeah, you just have to make sure you don't add any bubbles. So yeah, as I say, I'm more than happy if you start to set something up in a lab. More than happy to try and help. But yeah, that's pretty much it from me.
I have a question.
Yes.
We were trying to set up a sequencing lab, and we wanted to find out during sequencing, do you need to, how do you quality control? Because for every test, you find that there's a quality control, positive and negative control. So when sequencing, how do you ensure that how do you quality control the whole process?
So there are various steps that you could quality control. So in terms of negative controls, for the extraction, for instance, you might want to extract a water control and then run that through sequencing alongside your samples. And obviously, if you get reads of any bacteria or whatever, then there's the potential that you have contaminated it. In terms of positive controls, I guess if you've got H37RV in your laboratory for an extraction control, you could run that alongside it. Your extraction is, I guess, you'll see whether you've got DNA when you do your your QC for like a qubit and the gel electrophoresis. In terms of a sequencing positive control, yeah, again, you could run something like an E. coli if you have E. coli and extract that alongside it. And if you only get E. coli DNA, then that would suggest you're not contaminating things. I would imagine that if you're worried about contamination, then that would be seen in your other tests that you're doing anyway, especially if you're using sort of the same seed culture. But in terms of sequencing positive controls, it's not really that it's working or not. It will work. It's just that, as I say, it's more that you won't. It's the extraction that would potentially be the issue, I think. So having a negative control for that, and also I suppose, yeah, you wouldn't sequence anything, so you would know that hadn't worked because you can see how many reads you get for each barcode. Yeah, I'm not sure that's answered the question, but I'm not actually really sure exactly how. That's how we did it anyway. We ran a negative water control throughout the whole thing, and you could also use specifically bought DNA as well. So you can buy DNA from H37RV, and then you could run that alongside your sequencing run. So run that alongside your extracted samples. But sequencing in itself kind of is a validation because at the end of it, you will see what you get. So you can see that it has worked or not.
Okay, thank you.
Have a look at the website. Hopefully it will explain it. Have a look through the manual, because that is very, very detailed, and it should explain all of the bits. It also explains how the actual protocols for all of the different versions of the things that we did. So the normal CTAB and then the CTAB, the column-based CTAB. So yeah, I think I would read through that, and that hopefully will familiarize you with the process alongside watching the videos.